This tutorial will demonstrate how to use the KSIT simulation software to run a SNP microarray. First, some background information. SNP stands for single nucleotide polymorphisms, or in other words, a single base difference between two DNA sequences. In the example shown below, the two sequences are identical except at the position highlighted in red, where one sequence has a T and the other sequence has a G. These differences may occur in genes where they may have some functional impact on the protein that is translated from that DNA, or they may be in non-coding regions. But in either case, these SNPs can be associated with diseases or with other conditions. SNP microarrays use pairs of probes that each contain one of the two possible bases in that sequence. And those probes are spotted onto chips in an array pattern, and then a DNA sample that is labeled with a fluorescent dye is allowed to bind or hybridize to those probes. The fluorescence intensity, or the amount of fluorescent dye that binds to each spot, indicates the strength of binding to that probe. The ratio of the fluorescence intensities for the two spots is used to determine the genotype. Here's an example. We have two probe sequences that are identical except at that SNP position shown in red. The fluorescence intensities are shown on the right, and you can see that the amount of fluorescence binding to the second probe with G at the SNP position is much higher than the fluorescence intensity for the first probe. This indicates that the DNA sample has a better sequence match to the second probe, and therefore it must have the G nucleotide at that SNP position. So the genotype for this DNA sample would be called as homozygous for the G allele, or GG. If the fluorescence intensity was higher for the first probe, then the genotype would be called as homozygous T, and if the fluorescence intensities were approximately equal, then it would be called as heterozygous. More details about the mathematical calculations that are used to call the genotypes for a DNA sample and to compare the genotypes of different DNA samples are provided at the end of the tutorial. To run a SNP microarray using the KSIT software simulation, open the software as usual and then find the Chip button on the silver button bar. Click the Chip button and then select Open, and in this case we want to open and load a SNP chip file. Note that the alternative is to open an expression chip file, which is a different type of microarray. The files for the microarray cases are located in the cases folder, which is in the main folder that is extracted from the download. Inside the cases folder there is a separate folder for the microarray cases, and we are looking for SNP microarrays. Note that there's a single file for each of these cases, which is different from most of our other cases that require multiple files to be opened to analyze the case. These files are also in CSV format, so they're comma delimited text files, which is again different from the other cases that use standard text files. I'm going to open the SNP tutorial file, and these files contain all the DNA sample information and the probe information, as indicated by the orange spots here. Each chip has an array of just 64 probe spots. In an actual microarray there will be tens of thousands of spots, but we've focused on just 64 features to make it more understandable. In the Open and Process window, the name of the file that was open is visible, and then the samples that that file contains are listed below. And that will carry out all the steps involved in applying the DNA samples and detecting the fluorescence. So here are the results of a SNP microarray, and you can see that now the spots have fluorescence associated with them, and the spots have varying degrees of fluorescence intensity. The values for the fluorescence intensity are indicated in this grid over on the right, and there are 64 cells in this grid that correspond to the 64 spots, and the probes are associated in pairs, in the SNP pairs. If we just focus on these two cells here, they have the same SNP ID number. They're labeled A and B to indicate that they are two different probes in a SNP pair. Clicking on one of these cells will highlight the spot that is associated with that particular probe. The fluorescence intensity value is indicated also in the cell. And you can see that cell 1A has a lower intensity than cell 1B, and that corresponds to the brightness that you can see by eye over in the spots. Clicking on one of these cells also will reveal the probe sequence, which is down here in the lower right part of the grid. And the center base in that probe sequence is capitalized. That is the location of the SNP. And in this version of the probe labeled B, that base is a T. If we click on the A version of that probe, that is a C. And this particular DNA sample is binding more tightly to the probe with the T in the middle, the second probe, than it is to the probe with the C in the middle, and therefore the genotype is being called as homozygous for T. That means both chromosomes in this DNA sample probably contain the T version of that SNP. So the genotype is TT. The genotype call is written in the cell that corresponds to the probe that contains that base, and the call is determined by calculating a ratio of the fluorescence intensities for the two probes. And there's more information about the math behind this analysis at the end of this tutorial. But let's take a look at a different pair of probes down here in the bottom of the first column. The fluorescence intensities are relatively close, and if you view the spots, you can see that they appear to be the same intensity. Therefore, the ratio of those two intensities will be around 1, and that will generate a genotype call that is heterozygous. So there is approximately equal binding of this DNA sample to each of the two probes. This file contains 3 DNA samples that will be run on 3 separate chips, each with the same array of probes. You can navigate between the different DNA samples using the Open and Process window by clicking on the names of the samples. So we have a positive control, we have a negative control, and we have a patient sample. And as we click on those different samples, you can see that there is a change in the array of spots with the different fluorescence intensities, and that will correspond to changes in the data in the cells on the right. You can also navigate using the navigation window up above the main window. These thumbnails will be in the same order as the sample names in the Open and Process window. So I can click on each of these and move between the different samples. To know which sample you are viewing at any given time, if you look underneath the window on the right, it says this chip is the patient. So the chip currently visible, the results are for the patient DNA sample. This also indicates the last chip that was viewed. This is important because one thing you might want to do with these samples is compare them. Depending on the nature of the case and the case scenario, you might be looking at individual samples and just determining the genotypes at particular SNPs, which might be diagnostic or important for a research question. Or you might want to compare different samples and see where there are differences in the SNPs. To compare 2 DNA samples, first select the 2 samples that you want to compare. We can do that in the Open and Process window. Let's compare the positive control and the negative control. And then that will be indicated here under the grid where it says this chip is the negative control that we're currently viewing. The last chip is the positive control. And then to compare those 2 samples, use the chip button, select Compare, SNP chip, compare the last 2 SNP chips selected. Now you can see that in the grid there are, first of all, two sets of genotype calls for each pair of probes. The blue call corresponds to the negative control, which is colored blue at the bottom here. The purple genotype call corresponds to the positive control. For any probe where the genotype calls are different between those two samples, those probe cells are highlighted in yellow. So we can see for this particular set of probes, the negative control has a homozygous GG genotype, while the positive control has a homozygous AA genotype. And then there are two other SNP probe pairs where the genotype calls are different for the negative and the positive control. While we have the compare feature activated, we can change the DNA samples that are being compared just by selecting them. So we can go back to the Open and Process window and select the positive control and the patient sample, for example. And now we can see that there are differences between the positive control and the patient sample. They look very similar to the differences we saw between the positive and negative control. So if we compare the negative control and the patient sample, we see there are actually no differences between those two. So for this example, we can conclude that the patient DNA sample has the same SNP profile as the negative control. And so that might be diagnostic for this particular case, to be able to say that the patient is negative for the disease that is being tested. But for some cases, we might want to gain more information about particular SNPs, for example, the SNPs associated with the positive control sample associated with the disease. So if we go back to comparing the positive and negative controls, we can get more information about these particular SNPs, what genes they might be associated with, etc., by selecting either the SNP ID or the probe sequence and submitting those to databases where we can get more information. So if we wanted to investigate this particular SNP further, we can click on either cell because they both contain the same SNP ID, and the DNA sequences of the probes are very similar. They're only one base difference. So I'll just click on the first one and note that that highlights the spot associated with that probe. And then right-clicking on that spot pulls up a menu that allows you to either copy the probe sequence into the clipboard and open up the NCBI BLAST site to submit that probe sequence for BLAST analysis. Or you can select the SNP ID to be copied to the clipboard and open up the NCBI SNP database. Let's select the first option and open the NCBI BLAST site, and the BLAST nucleotide search page will automatically open in your default browser. And in the box where you would input a sequence, you can just right-click and then paste, and the sequence that was stored from the SNP microarray for that particular probe will be pasted in. And the default parameters are set so that it's not just looking at human DNA, it's looking at all DNA. We can leave that the way it is. We can use the BLAST-N program to look for somewhat similar sequences. You may want to change some of these parameters, but I'm going to leave them at the default settings and then click the BLAST button. And this will then take that sequence and match it up against all of the other nucleotide sequences in the NCBI database. And here are the BLAST results. This is not a tutorial that will go into a lot of detail about BLAST and how to interpret BLAST results, but for purposes of the SNP probe analysis, what I want to point out is that when you look at the list of matches, they may or may not be in genes. Many SNPs are not located in genes. They're in non-coding regions. And so the kinds of sequences that they would hit are clones of DNA, back clones, other kinds of clones. So it's not always particularly informative in terms of understanding what the function of that SNP might be. But if a SNP is located in a gene, then you would be able to click on the GenBank accession number and perhaps get some more information, access to publications about that SNP. So instead, let's look at another type of database, the SNP ID database, which is also on the NCBI site. This option will save the SNP ID into the clipboard and automatically open up the NCBI SNP database page. You can then, again in the search box, right-click and then paste, and that will paste the ID number into the search window. Since it's stored in the clipboard, if there are other databases on other websites that you might want to search, then you can use that same technique of pasting the ID into the search window. Conduct the search. Clicking the SNP ID name takes you to the report page for that SNP. And again, this is not going to be a full tutorial on how to use the SNP database, but I'll just point out a few features that can be informative. There is no clinical significance that has been reported for the SNP, so if that's what you're looking for, this might not be the SNP to pursue. But if we look down the rest of the page, there's some information about how it's been tested previously and in what populations. There's a gene view that indicates that this gene is on chromosome 12 and where on chromosome 12 it is located. And then you can see if it's nearby any other genes. So there is a gene located not too far away, and you could then get more information about that particular gene, and if that makes sense in the context of your case, that might be worth pursuing. Again, is a list of the different types of microarrays that incorporate this particular SNP, some allele frequency analysis in different populations. So again, depending on the nature of the case, this might be important information. Let's look at one more example to view a different type of result. Here's another SNP difference between the positive and negative controls. So if we click on that and then right-click on the spot, copy the SNP ID to the clipboard, and open up the SNP database. And we can paste in that SNP ID and search. And if we click on the SNP ID, the report page indicates that there is a source, at least to the SNP. That indicates that the SNP has been associated with intracranial aneurysm and coronary heart disease. So if that's the nature of the case that you're investigating, then you can get a lot more information by pursuing these references. The last part of this tutorial will focus on the calculations used to determine the SNP genotypes for different DNA samples. And for each SNP probe pair, it is the ratio of the fluorescence intensity for those probe spots that is used to determine the genotype. So first of all, this is what the data files look like for the SNP microarray cases. These are CSV files that can be opened in Excel, and the first column is the SNP ID with A and B indicating the two different versions of the probes. Here are the probe sequences, with the center base being the SNP variant, and then each DNA sample has a column of data indicating the fluorescence intensity associated with each probe. And again, it's the ratio calculated by taking the fluorescence intensity for the second probe and dividing it by the fluorescence intensity for the first probe that determines the genotype. And let's look at a different format for an Excel file that shows those calculations. Here's another spreadsheet that shows the formulas used to calculate the fluorescence intensity ratios and determine the genotypes. So this has the same column of SNP IDs and probe sequences, and remember that the SNP probes are in pairs labeled A and B, representing the two different versions of that SNP, the two different alleles. And This spreadsheet again contains the formulas. So there are two sets of fluorescence intensity data for two different DNA samples, just two at a time since that's how we compare them. And then for each one of those DNA samples, a ratio can be calculated for each probe pair. And the ratio is calculated simply by taking the intensity value for probe B and dividing it by the intensity value for probe A. And that formula is shown up in the formula line. So in this case, probe B yields a much higher fluorescence intensity than probe A, and so the call for that genotype is BB. And this spreadsheet uses just a generic A and B for the two different alleles that is not obviously related to the actual nucleotides at that position. If the two intensities for a probe pair are relatively close together, then that will yield a ratio around 1. And if the calculated ratio is between 0.5 and 1.5, then a heterozygous genotype will be called. And finally, any SNPs where the two DNA samples have a different genotype can also be determined mathematically by calculating a second ratio, taking the fluorescence intensity ratio for the second DNA sample and dividing that by the ratio for the first DNA sample, and that second ratio or meta ratio is converted into a log base 2 in order to amplify the differences. And it's the absolute log, so either negative or positive values will be detected. And for any second ratio where the absolute log 2 value is greater than 2, that will be scored as a difference, and the cells in the simulation will be highlighted in yellow. And these are the same data that were used in that simulation, and as we scroll down we can see there are 3 SNP pairs where the overall ratios are different, and so those are scored as differences in the genotypes. And if we look at the genotypes, we see that for each one there's a different call for DNA sample 1 versus DNA sample 2. And this spreadsheet is included with the case files, with the tutorial files, and so if you wanted to see these calculations for the data for a particular case, you can replace the columns, the SNP ID, the probe sequence, and the fluorescence intensity columns with data from that case file.